mesothelioma, so if you need to check your email, check your Facebook page, so this may be a good time. I'll touch a little bit on KRAS and lung cancer, but I think, okay, so let's see. Okay. So most of the patients, as you know, with mesothelioma are present with advanced disease and are not candidates for surgical resection. You just need to look at those scans, and really surgery is really not an option. And the only FDA-approved therapy is combination of pemetrexed and cisplatin. So that's the standard of care. So if you are a medical oncologist, that's all you need to know. And that was based on this pivotal trial by Nick Vogelsang, which showed that combination of pemetrexed and cisplatin compared to cisplatin resulted in a median overall survival of 12.1 months. So this is the standard of care. The drug was approved in 2004. So has there been any progress since 2004? So surprisingly, there have been many well-designed large phase 1, phase 2 clinical trials in mesothelioma, and there has been a lot of interest both by academics as well as by industry. But unfortunately, most of these studies have been negative. However, there are some promising therapies, and I will just talk about 2 or 3 of them, and I will talk about the immune checkpoints later on in the program. So most of the single-agent anti-angiogenic agents such as sorafenib, siderinib, sunitinib have been negative. However, there are 2 agents which show some activity when combined with chemotherapy. This includes bevacizumab and nintedanib, a multi-kinase inhibitor. So this was a study that was presented at ASCO last year. This was done by the French group. This was a randomized clinical trial where patients were treated at a standard dose of pemetrexed and cisplatin for 6 cycles, or pemetrexed and cisplatin with bevacizumab, followed by bevacizumab maintenance. So this was a large study, and it showed that the combination of pemetrexed and cisplatin resulted in a median overall survival of about 18 months. But as you notice that the median overall survival of the control arm in this study was about 16 months. It's about 4 months more than the Vogelsang study. So it could be because of changes in the standard of care over the last many years, but clearly the control arm OS was much larger than you would expect. So as of the last I heard, that Roche was initially going to file for registration, but I think they are not doing that. This was not a registration trial. So the future of bevacizumab in clinical practice is not clear. I don't think it is used as often, at least in the United States, and there are hurdles in other places. The other anti-angiogenic agent which has recently shown some activity is nintedanib, and this was paper published earlier this month in JCO. So this was a randomized controlled trial where the patients were treated with chemotherapy, pemetrexed cisplatin, or pemetrexed cisplatin and nintedanib. Again, the number of patients are small, about 40 patients in each arm, but the PFS is clearly looks very impressive, but I think the part of it could be is because of the small number of patients on the study. And the overall survival was not statistically significant, but this has led to a larger trial called as the LUM-MESO, which is a global randomized phase 3 study. So it's actually a part of the same trial. It's a phase 3 component of it. So we'll have to see whether this really results in improvement in OS in a larger population. So what I'm going to talk mostly in the next 5 minutes is mesothelin targeted therapies for mesothelioma and hopefully for other common cancers. So mesothelin is a cell surface protein that was discovered in Ira Paston's lab many years ago, and we called it mesothelin because it's present on normal mesothelial cells. And this is a protein that I started working on in 1994 when I joined Ira's lab as a fellow. And we showed that it was a new target for immunotherapy, and recently we have summarized our work on mesothelin. So what is mesothelin? It's a cell surface glycoprotein which is attached to the cell membrane. It is processed to a shed fragment called mesothelin and another fragment called megakaryocyte potentiating factor. And you can detect both of these proteins in the blood, and there could be potential markers for activity. The most important thing is that its expression on normal human tissues is limited only to the mesothelial cells. And this is critical because the mesothelial cells are dispensable. You do pleurodesis and things like that. You can damage them, but they're not essential for survival. So that's what makes it an attractive target for therapy. And we and others have shown that mesothelin is highly expressed in many cancers, almost all epithelial mesotheliomas, about 80% of pancreatic cancer, ovarian cancer, and about 50% of lung adenocarcinomas, as well as other tumors, including thymic carcinomas. So mesothelin-directed therapies as a proof of concept, if they are successful, could be useful for treating many common tumors. And our group at NCI is right now focused on pancreatic cancer, and we're starting to get into the lung cancer field with mesothelin-targeted therapies. So just to show a paper that we published a couple of years ago showing that there are different patterns of expression in lung adenocarcinomas, with about 50% of the lung adenocarcinomas having some expression and about 25% having good expression, which we defined as more than 50% expression. And we also showed that KRAS mutations are present in the majority of the patients with high mesothelin expression, which we defined as more than 50%. About 70% of the mesothelin positive tumors have KRAS mutation, and it is mutually exclusive with EGFR. None of them have EGFR mutation. So there's a lot of interest in KRAS mutations, lung cancers targeting the mesothelin, and there are a couple of Stand Up to Cancer grants for developing different therapies for KRAS mutant lung cancer using mesothelin therapy. So there are several mesothelin targeted agents in the clinic, and this is just an overview that we published last year, including immunotoxin. This was the first molecule ever developed to target mesothelin. We did a clinical trial, treated the first patient in 2001. We have developed another molecule which is reduced immunogenicity called LMB100. That's in clinical trials at NCI. The folks at Bayer developed an antibody drug conjugate, inutumab. There are also a couple of other molecules. These are Listeria monocytogenes expressing mesothelin. So there's a trial in mesothelioma with CRS207, and there's an ongoing trials in lung cancer using a similar molecule in J&J compound. And in addition, there are several clinical trials going at NCI Memorial and the Carl June's group at Penn developing CAR T cells for mesothelin. So my work predominantly over the last couple of decades has, along with Ira, focused on immunotoxin. I know immunotoxins were used many, many years ago and they were given up, but I think we have learned a lot over the last many years. So an immunotoxin consists of an antibody or an antibody fragment, usually an Fv linked to a toxin. So the toxin that we work on is made by the bacteria Pseudomonas aeruginosa, and you make it by a recombinant mechanism. And they have a unique mechanism of action that they kill cells by inhibiting protein synthesis. So this is very different from any other drug currently in the clinic, which either damage the DNA or have other mechanisms. So they could potentially be useful for drug-resistant tumors. So as I mentioned, SS1P was the first anti-mesothelin drug in the clinic. We did a phase 1 study. We did not see any objective responses, but the main reason was that it was immunogenic. So patients developed antibodies to the drug after 1 or 2 cycles. But the most important finding of this trial was demonstrating that it is safe to target mesothelin. So there was a lot of concern when we started the phase 1 that you would get horrible pericardial toxicity or death. But I think this trial established the safety of targeting mesothelin, and that led to a wide interest by many different groups to develop mesothelin-targeted compounds. As I mentioned, in this phase 1 trial, we did not see much activity because patients developed antibodies. So our efforts at NCI are focused on overcoming this problem. So we showed in our preclinical model that if you use pentostatin and Cytoxan, you can decrease antibodies to SS1P in mice. So we did a small study and showed that you could decrease antibodies in patients. If you gave SS1P alone, about 90% develop antibodies. if you gave it with pentostatin Cytoxan, you can significantly decrease this response. And we did see some meaningful response. This was a patient who got 6 cycles of therapy and had a pretty amazing and durable response. And she died recently, more than 5 years from treatment. But with this era of immune checkpoints, when you use pentostatin and Cytoxan, you suppress the immune system for about 6 months to a year with the decrease in the B and the T cells. So this strategy is really not feasible. So we have focused a lot of effort to develop a better molecule. And this is work done mostly in Ira's lab, where we have made a new molecule which is less immunogenic by getting rid of part of the toxin, identifying the human B and T cell epitopes, mutating them. So we have developed this molecule called RG7787. It has reduced antigenicity, and my lab has shown it's much more active against mesothelin-expressing cells. So we currently have an ongoing clinical trial for patients with mesothelioma. So the other anti-mesothelin drug which is in the clinic is called enitumab ravtencine. So this was developed by the folks at Bayer. So it's an ADC. It consists of an anti-mesothelin antibody linked by a disulfide linker to the metasenoid derivative DM4. It's a tubulin inhibitor. And the linker is pretty good. Yesterday we had some discussion about ROVA-T. So in this one, there is very little free drug that is present in the circulation. So we've shown that. And in the phase 1 trial, it showed activity with 5 of the 16 patients having a partial response. So this trial went on to a randomized phase 2 study as second-line therapy in patients with pleural mesothelioma. So it was a randomized study, patients getting enitumab or getting navalbine. And so these were the results that were presented at World Lung, that there was actually, the PFS was the primary endpoint. The PFS was very similar. But if you look here, that our assumption was that the PFS of navalbine would be about 2.5 months, because that's what was published in small studies. But clearly, navalbine by itself has results in a PFS of 4.5 months. So there was no difference in the PFS. We'll look at the measured data. But this is a very important study in drug development in mesothelioma because it establishes a benchmark for second-line PFS in mesothelioma. So this is the largest trial of navalbine multi-institutional study with about 80 patients getting navalbine. However, enitumab is in clinical trials in many different indications. In a basket study that's ongoing, we do have a clinical trial in lung cancer for enitumab. We're also doing a clinical trial of enitumab with PAM-CIS. This was results presented late last month. So it looks like in the small number of patients, we see increasing responses, patients receiving PAM-CIS with enitumab. So we'll have to see where this drug goes, but I think it might be dependent upon the target indication. And lastly, there are trials with CRS-207, which is a live listed monocyte genes expressing mesothelin. So there was a lot of concern when we did a phase 1 trial about injecting bacteria in patients. So we had to go through a lot of hoops, but it is safe. And this was a study that we published last year, presented last year, combining CRS-207 with pemetrexed and cisplatin, results in an overall response rate about 50 to 60%. So this molecule is currently in clinical trials with pembrolizumab as second-line therapy for mesothelioma. So in summary, in terms of mesothelin-targeted agents, the anti-mesothelin immunotoxin LMB100 is in clinical trials for mesothelioma and pancreatic cancer at the NCI. Enitumab ravtencine is in clinical trials with chemotherapy, and there will be a trial with pembrolizumab opening up shortly. CRS-207 is already in clinical trial as second or third line therapy with pembro, and as I mentioned, there are trials of mesothelin CAR T cells. Many of these are focused on pancreatic cancer, mesothelioma, but the PEM group is opening up clinical trials for longer term carcinoma. That's it.